So good morning, everybody. 2 years ago, I got the invitation to give a lecture at the Dutch Neuroscience meeting in London, and I think most of you actually know this meeting. And the title of the session that I was supposed to talk in had the title, The Cerebellum, Who Cares? And when I first read the email, I thought, no, this must be a joke. But then, I read further in the email, I recognized that the organizer really had the attention or the idea to draw attention towards the cerebellum and say, please come to our session. It's an important part of our brain. And I think this meeting has a few hundred participants. And indeed, who cared? Only a dozen or so showed up at our session. So in the end, I don't know if it was the title that really scared them off or the quality of the presenters, but we will never know. But it's really true. If I go through programs of scientific conferences, you will find sessions that cover literally every part of our human brain. But there are hardly any sessions that cover specifically the cerebellum. And that is really disappointing me always. And this is also reflected in the number of hits when I type in cerebellum in PubMed. You hardly reach 80,000 papers. Whereas if you type in hippocampus, this little tiny organ, also a nuclei in the brain, but linked to a big disease, you hit almost twice as much numbers. And so I've been in this field working on the small brain, on the cerebellum, for over 15 years. And I still have no idea why the cerebellum is not of great matter to the scientific community. And I really have no idea, because please take a look at it. It's a magnificent organ. It looks beautiful. It looks like a tree. Not only from the outside, but also from the inside. It's a small part of our brain. It only accounts for 10% of the total brain volume, but it contains over 50% of the total neuron count of our human brain. So it must be very important. So not only beautiful from the outside, it is perfectly folded in the way that it is, but also from the inside, the cells that it contains have a highly organized manner. So for example, we have the Purkinje cells, the key regulators of our cerebellum. These big cells with their extensive dendritic trees that are being innervated by parallel fibers from granule cells that all move very orderly from right to left, from right to left. So each Purkinje cell is innervated by hundreds of thousands of parallel fibers of these granule cells. And the other way around, each Purkinje cell is disconnected to hundreds of thousands of granule cells and the parallel fiber. And from the inferior olive, you have these climber fibers coming up these Purkinje cells tree. So you can envision this as a unique neuronal circuitry that has a very delicate communication circuitry, and any disturbances in that can have detrimental effects. But actually, what does the cerebellum do? So I think that most of you that came home after a party and had too few drinks knows what the cerebellum does when it doesn't function properly. But I think the best example is actually this. Here you see a superb cerebellum in action. This is the gold medal winner, Sanne Wevers, with her balance beam performance. And her cerebellum is working optimal. She can stay in balance on this 10 centimeter width balance beam. She doesn't drop off. The way that she can control her movement on this small balance beam is perfect. And the fact that she's able to learn how to perform these accurate and smooth movements. This is what we call motor learning. So these are all aspects of a proper working cerebellum. And the cerebellum doesn't work alone. It is an organ that communicates. It receives information from the inner ear, it receives information from the vestibular system, and it also receives information from the sensory motor system as well, by which it adjusts the smoothness of the movements. It even can calculate the speed of movement and then adjust the motor command accordingly. So this is a very clever organ. It participates in this circuitry of information that all controls our body movement. So it's not the generator of motor commands, but it's actually the fine-tuned machinery that helps us to have a fluent and easy and easygoing movement of our body. So the cerebellum does way more than this. performing and controlling motor commands. It also has an important role in cognitive functions, in emotions, and in language. But that will be not part of the talk today. So actually, the first time that I encountered an individual with a cerebellar deficit was at the yearly patient meeting of the Dutch Ataxia Society. I was a first-year PhD student, and I really had no idea what to expect. There I saw all these individuals that needed aids to walk or keep their balance up straight, or were even wheelchair-bound. And that really had a huge impact on me. I was quite nervous to give the presentation. I had to talk about my genetic findings. I was already studying the genetic basis of this very rare neurodegenerative disease called spinocerebellar ataxia. And these patients they were suddenly sitting in front of me, and I had to tell them, about their genes, about their genetic deficit. That really had a huge impact on me. So these patients, this disease is caused by pervasive Purkinje cell loss in the cerebellum that only occurs at late age of onset. That's an interesting thought, that you can live your life healthy up to 40, 50 years of age without any complaints, and then suddenly the disease symptoms start to appear. And this is really a burden for the patients and their relatives, as it often interferes also with family planning. So at this meeting, I presented my first genetic data. This is a genetic disease. It's inherited in an autosomal dominant fashion. That means that if you're a carrier of the mutated gene, 50% of your children, of your offspring, will also develop the disease at a later age. So this is what we call a genetic heterogeneous disease. It's not one disease. And this is really a puzzle for us geneticists, clinical geneticists, and neurologists. To be able to solve this puzzle, we need to understand the complexity of the cerebellum. We need to unravel all these genes, find all those genes and the associated biological pathways that make these Purkinje cells more vulnerable for a genetic insult than all the other surrounding neurons. We still have no idea why that is. And how it comes that if you have 40 different disease genes, because this is the number that my team and others have been discovered so far, that 40 different disease genes, when mutated, can all lead to the same disease phenotype. There are no 40 roots to cerebellar ataxia. Probably there are shared mechanisms. And we try to solve that as well. Can we identify those shared mechanisms? And yes, we can. Recent data has shown, based on our novel genetic studies, that the new genes that we have picked up highlight pathways involved in synaptic transmission and in nuclear function. So these are routes that we are going to exploit in the coming future. So my team made it a mission to provide a genetic diagnosis for these patients. And despite the fact that we already know 40 different disease genes, we only can explain 60 to 70% of these cases. The rest remains genetically unsolved. And that is also one of our missions, to do the gene hunt. And the fact that it still remains unsolved is not because of the state-of-the-art technology that we have currently, because we have now this next-generation sequencing technologies. We can sequence whole exomes, we can sequence whole genomes, we can even do the RNA-seq, and this will allow us to get to a genetic diagnosis actually in a few weeks. But this is not the case this is not in all cases leading to a positive diagnosis. And the reason for that is actually due to several reasons. One can be that the technology does not read all parts of our exome or genomes as good as it should be. So think, for example, about repetitive sequences in our DNA, copper number variations. So these are bottlenecks of the technology. It cannot handle those. We can have mutations in our mitochondrial DNA, whereas not in the nuclear DNA, and that has not been captured by this technique. It can even be that we have mutations in microRNAs or in long non-coding RNAs. And I find that actually a very interesting thought, that we can have mutations in this regulome that may cause a genetic Mendelian disease like spinocerebellar ataxia. And this interesting thought we will explore in one of our upcoming projects. But actually the main reason why in 30% of the cases we cannot have a genetic diagnosis is that for most of the genetic variations in our genome, we have no idea what it functionally does. We literally have no clue. There's so much genetic variation out there with an unknown function. And this is something that we clearly have to solve. How do we interpret these variations and how can we classify them in a proper way as being pathogenic or classify them as rare but benign variants? And to explore this, we are currently using alternative technologies like RNA-seq, in addition to the DNA analyses. So this technology allows us to unravel the transcriptome. It will highlight the genetic variations in the transcripts, but it also will assess at the same time the gene expression levels. And when we think about functional deficits, we generated the hypothesis in our team that if a variant is likely to be pathogenic, it might affect gene expression levels. That can be in a gene nearby, but it can also even affect a gene that is somewhere on even on a different chromosome. And thereby it does not allow us the direct output or the direct functional consequence of this particular genetic variant, but we can also assess the downstream consequences. We can unravel the biological roots that are affected by a particular variant on a particular chromosome, and by detecting the alterations of gene expression variations somewhere in our genome. So this technology is working quite well for diseases that are not brain linked. So here's the main challenge. Can we use this technology to assess blood expression levels, to determine the pathogenicity of a variant that leads to a brain disease? Well, this current challenge we're currently exploring. And we have some very promising hopes that indeed a disease or a variant that causes a brain disease can leave a fingerprint in the blood. So besides all this bioinformatics and genetics, my team heavily relies on in vitro models and in vivo models to really study the functional consequences of particular genetic deficits. So we make use of neuronal-like or primary neuronal cell cultures in which we overexpress our putative disease genes and analyze the functional consequences using specific readouts that are specific for each different gene. And that works really well. However, there is a pitfall of overexpressing disease proteins, if then you have the disease protein in excess, and that doesn't really mimic the endogenous situation. So we're currently using this genome editing technology, the CRISPR-Cas9 technology, to introduce the human mutations in the genome of cells, but even in mice, to mimic the more disease-like status in an endogenous situation. And so far, it's looking pretty successful. With respect to the in vivo models, we're using Drosophila, in collaboration also with ODC Bonn, and we're using mice. We had failures and we had successes. Luckily, the successes are the ones that you remember best. So we have a beautiful model mimicking spinocerebellar ataxia type 23 in the mouse facility here at the UMCG. And this mouse model really allows us to directly analyze the effect of a disease gene, a human disease gene, in vivo, and study the effect on cerebellar functioning, but also on cerebellar pathology. And this mouse then allows us to test for compounds that can reverse or stop or halt the disease symptoms from occurring, and also the cerebellar deficit that we see in these mice. And in the end, we hope, of course, that it may lead to the opening up for therapeutic development for drugs that can treat these patients, as these are still incurable diseases. So in the end, I hope that I've convinced you that the cerebellum does matter. We should actually appreciate the cerebellum a bit more. The fact that I'm standing up right here and talking to you right now, or that we cycle home in the sun after this great meeting. Just simple facts that we take for granted, but which are not simple things for an individual that suffers from a cerebellar deficit. So I really think the cerebellum does matter, and it's definitely worthwhile investigating. So thank you for your attention.